In this video, I'm going to tell you about transcription, which is the first part of what we've defined as the central dogma, or DNA to RNA to protein. So transcription is the steps where DNA goes to RNA. So the first thing we might ask ourselves is, why does the cell even need to do transcription? What is the point of the central dogma? Why does DNA need to go to RNA, need to go to protein? And the reason that we need to do transcription is that all of the genetic material of our cell, all of the DNA, lives in the nucleus. It's in the nucleus in the eukaryotic cell. It's there so that it's protected from insults within the cell, so that it's all in one place and can be controlled. But when we want to make a protein, we need to take that DNA and use what's in the DNA to make the protein. And that piece of DNA that we use to make protein is called a gene. A gene is the term we use for a coding unit for a protein. And you can almost think of a gene as a recipe for making a protein. So all of our DNA is a cookbook, and it's full of recipes for making protein. So the next problem we have is that all of this cookbook, all of these recipes are maintained within the nucleus, just as I said. But all of the proteins are made in the cytoplasm of the cell. All of the proteins are made outside of the nucleus. All of the DNA stays inside the nucleus. So there needs to be a system that allows you to utilize these recipes out here in the cytoplasm while leaving your original copies of the recipes alone. And that system is mRNA, or messenger RNA. So the proteins are made in the cytoplasm, DNA stored in the nucleus, so the cell makes these copies, these mRNA copies, to be used elsewhere. So the formation of these mRNA copies is known as transcription. Later on, we'll use these mRNA copies for translation, but for now we're just focused on the DNA to RNA transition, which is called transcription. So there are several differences between DNA and RNA, and these differences are what makes this entire process possible and helps this entire process along. The big difference that's most obvious is that DNA is long. There's long strands of DNA all throughout the nucleus. mRNA strands are relatively short. They contain just one or a few genes, and the cell has a lot easier time working with just a single recipe rather than an entire cookbook. The second difference is that DNA is double-stranded. You'll remember that this is part of the function of how DNA replicates. It has two templates. It can make two new copies anytime it replicates. RNA, on the other hand, is single-stranded. And the reason that it's single-stranded is that it's more useful later in the process of the central dogma, when we get into translation, to use this single strand of RNA to make a protein. The other difference between DNA and RNA is that DNA is comprised of the sugar deoxyribose, which makes it very stable. Now, you don't need to worry about the chemistry that makes deoxyribose more stable than ribose, which makes up RNA, ribonucleic acid. But ribose makes the RNA molecules not as stable. Now, you can understand easily why DNA needs to be stable. The DNA is preserved in the cell. It contains all of the instructions that the cell needs to live. You want this molecule to be perfectly stable and be maintained generation after generation. The instability of the RNA molecule is actually important because you make copies of RNA to make proteins, but the cell also needs the ability to get rid of these copies. These copies need to go away, they need to degrade over time so that the cell isn't constantly making protein. So these RNA molecules are made more frequently. They're not as stable as DNA molecules. A good example to think of, you can consider the DNA as that cookbook. It's a bound book that you've got on a shelf. The RNAs are just photocopies of pages. Those photocopies of pages are cheap and easy to make. They rip, they tear, they get things all over them, and you go back to the original to make a new copy. The final difference between DNA and RNA is that the pairing rules slightly change. Now, you'll remember from DNA that adenine always goes with thymine, and guanine always goes with cytosine. These are the pairing rules you've learned, A to T, G to C. There's only one tiny difference when you get to RNA. you'll see that guanine to cytosine stays the same, but now adenine pairs with U for uracil. There's no thymine in RNA, there's just U for uracil. So when you're doing transcription and you see an A, instead of putting a T there, you're going to be putting a U there. So A goes with U and G goes with C. These are the major differences between DNA and RNA, especially those that are important to transcription. So how does transcription actually happen inside of the nucleus? And if you'll recall what we talked about with DNA replication, the process of transcription is actually very similar. You have double-stranded DNA here, and I've marked a 5' and 3' end, a 3' and 5' end on the DNA. And what happens for a copy, for a transcript to be made, is first, there's a sequence at the beginning of a gene called a promoter. A promoter is just a sequence of nucleotides that says this is where a gene starts. There are promoters all throughout your genome. Scientists are still trying to figure out how all of these promoters work, but they do have a good handle on a lot of them. And what these promoters do is they send out a signal for a lot of other components to build up on this part of the DNA. And the most important thing that builds up on this part of DNA is called RNA polymerase. Now, this is an enzyme. Remember, it ends with ACE. So this enzyme makes polymers of RNA. And it does this just like the DNA polymerase that we've talked about. It sticks down near the promoter and moves 3' to 5' along the DNA molecule. As it moves in this direction along the DNA molecule, a new RNA molecule is being made in the direction of 5' to 3'. So remember, these strands are anti-parallel. This molecule of RNA polymerase is zooming along, and as it's doing that, it's creating a new RNA right here in red. Now this RNA is going to be very much shorter than the DNA molecule, and this polymerase will continue along until it reaches a point in the DNA where it can no longer continue, for example, the edge of this replication fork, or sometimes it just eventually reaches the end of how far it can go with the tools that it has, and it falls off. So these things are constantly going and falling off. The neat thing about transcription as compared to DNA replication is that more than one RNA polymerase can sit down on this promoter. All of them can be going and making copies at the same time. So you have a promoter, that's where the gene starts. An RNA polymerase sits down near that promoter. It starts transcribing an RNA molecule as it moves 3' to 5' on the DNA, making a 5' to 3' messenger RNA, mRNA. We call this messenger RNA because the RNA is a message for another part of the cell. So the original process of transcription is relatively easy. The RNA polymerase sits down on the DNA, it creates a long mRNA strand that the cell can use later for translation, which we'll have in another video. But there is one more step that happens in eukaryotes, and this step is somewhat complicated. It actually confounded scientists for a while because they couldn't understand what they were looking at. But what your DNA actually has is it has long genes that contain actually more information than is required to make a gene, to make a protein. And so if you look at a mRNA, this would be called a pre-mRNA here. This is still in the nucleus. It has two components. Some components here in red are called exons, and some components here in green are called introns. And so a pre-mRNA is made of exons and introns. Exons, I always remember, are the expressed parts of the gene. So exons are expressed. Introns are not expressed. Introns are just pieces of RNA that sit between the exons that may contain regulatory elements, but by and large, scientists don't know exactly what the introns do. The introns are just areas within the gene that don't seem to make protein, don't seem to code for protein. So what needs to happen is this pre-mRNA gets processed before it leaves the nucleus so that all of the introns are removed and the exons are just put together into one red line like you can see here. So once this pre-mRNA leaves the nucleus, it's now an mRNA that's made of only exons, and we call this processed mRNA a coding sequence, or CDS, because this is now ready to code for a protein. Everything is set up. There's two other minor details I just wanted to point out to you. As this mRNA gets processed, it has a methyl cap added to the end, a methyl cap or a GTP cap. That's added to the 5' end. That helps move this molecule out of the nucleus through a tiny hole in the nucleus known as the nuclear pore. The other thing that's added to the 3' end is called a poly-A tail. It's what it sounds like. It's a lot of adenines, a long string of adenines that's added to the end of this tail. And the reason this is actually added is, recall that I told you that mRNA is much less stable than DNA. And the way mRNA degrades is that it degrades from the 3' end. Exonucleases start eating away at the 3' end. So what the cell does to buy itself a little more time, to make this mRNA last a little bit longer, is that it adds some extra A's to the end of every mRNA it makes. So even as an exonuclease, an enzyme that digests RNA, starts chomping at the 3' end, it takes a while before it gets to the red part, which is the important part. The last detail to point out when you look at all these exons is that scientists have found in many cases that exons can be arranged in different orders. So even within the same organism, the same gene can be arranged in two different ways, by leaving an exon out, adding an exon in, rearranging an exon. And these are called splice variants, because exons are being spliced together during processing. And if you do that in a different way, you get splice variants. The neat thing about splice variants is that a cell can take one gene and get several different proteins from it. So it's a useful a way of maximizing the cell's resources. So finally, just to tie a lot of the ideas together that we talked about today, transcription is the first part of the central dogma. Transcription is the step where we go from DNA to RNA, and specifically we go from DNA to mRNA, messenger RNA. But as long as you remember DNA to RNA, you'll know what transcription is. The mRNAs that are made from transcription are short. They're much shorter than the full strands of DNA. They're less stable, and they're copies of genes. So they're copies of genes that will be used later to make a protein. The pairing rules when mRNAs are made during transcription are different. G to C stays the same, but now adenine pairs with uracil. So A to U, G to C. When you actually get into the process of transcription, what happens is that the DNA strand opens up. There are sequences known as promoters. These promoters indicate the start of transcription, a place where transcription should start. And at this promoter, RNA polymerase, which is the enzyme that polymerizes RNA strands, is the critical enzyme that makes the RNA. It moves 3' to 5' while creating a 5' to 3' mRNA. The last piece you need to know is that these mRNAs are then processed. The mRNAs, the pre-mRNAs that are made during transcription contain introns and exons. The exons are the parts that are expressed. They need to stay. So the introns need to be removed before the mRNA leaves the nucleus. Once these mRNAs are floating throughout the cytoplasm of the cell, they can then be used to make proteins, which is the subject of another video.